All right, so today we are going to be discussing the topic of diffusion and osmosis. And this is going to be the first lecture in what will be our new module over cell processes. And so this is part of a larger unit that will talk about transport and transporting things across the membrane. So we'll also talk later about protein transport, protein-mediated transport, and then we're going to also discuss vesicle-mediated transport. And those will be the 3 days of material that we'll talk about concerning transport. And then as the series goes on, we'll also be covering cell communication and cell division. And those 3 major topics will form what we call cell processes. So this last module focuses around cell processes, starting today with the process of cellular transport. And so cellular transport, to start talking about this, we are going to draw a membrane across the board. And so we'll label this as a membrane or a phospholipid bilayer. And the phospholipid bilayer itself has these polar heads, and then we've got the hydrophobic tails. And what happens is we describe this membrane as being semi-impermeable. Or you could also say that it's semi-permeable. And that just means that there are certain molecules that are able to go across the membrane all by themselves, and other molecules that are not able to go across the membrane. And so if we start to think about what kinds of molecules might go across the membrane, on this side, some molecules that freely go across the membrane are going to be some molecules that we just talked about in our last unit, oxygen and carbon dioxide. Notice as we went through that unit, we talked about these 2 molecules, and we didn't talk about any transport processes for them. And that's because both carbon dioxide and oxygen, molecular oxygen, can just diffuse right across the membrane. They are permeable to it. Then on the opposite side, we'll kind of draw it over here and make kind of a scale. We have things like DNA and RNA, or proteins, or even things like glycogen or starch. And these ones are completely impermeable. They won't go through the membrane at all. And then we have this kind of range in the middle of things. So if we were to add some other things up here, we might put down things like ATP or amino acids. We might have something like a phosphate ion. And then we might have ions, for example, a chloride ion. We might have sodium ion, potassium ion. And what we find is all of these things are impermeable. We'll call them essentially impermeable to the membrane, meaning they don't go across. And the reason that they don't go across is because of their properties. Over here, these things are very large, and so they're much too large to break through the phospholipids. They would displace too many phospholipids. And over here, these ones, chloride ion and sodium and potassium, are actually quite small. In fact, it'd be hard. These are single elements, single atoms. It'd be harder to get smaller than these items. However, these are charged. And when you have a formal charge on the ion or the molecule, the atom, we find that it's really quite impermeable to the membrane. They just don't go across. And so the properties, if you are large and or charged, remember that DNA, because of the phosphate groups on it, also has a negative charge on it. Proteins are often charged. ATP, we discussed in our last unit, has a bunch of negative charges on it. Amino acids will have positive and negative charges. So if you're charged and or large, then you're going to be impermeable to the membrane. Over here, the properties, when you look at things that are completely permeable to the membrane, you're going to find that these are small and they are nonpolar. So both oxygen and carbon dioxide are nonpolar. You might also add to this group if you had something like methane. So CH4, that would be pretty permeable to the membrane because, again, it's small. It's only 5 atoms, and it's nonpolar. Then you have some other molecules in here. Let's draw, for example, we just talked a lot about the molecule glucose. Still fairly small, although this has a molecular weight of about 180 daltons. But you'll have glucose. You might have something a little bit farther on this scale. Let's take a lipid like estrogen. And let's take another real small molecule like water. And these ones, these molecules, right in here, we have what we call a limited permeability. And then these molecules would be freely permeable. And so there's actually a scale or a continuum of what goes across the membrane. And if you were to look at this in the limited permeability area, glucose would be less permeable than, say, something like estrogen. Why? They're both relatively close to the same size, but glucose is fairly polar. Estrogen is more nonpolar, so it's going to be more permeable to the membrane. Over here, water, we've learned earlier, is actually pretty permeable. So why did I put it down here on the more permeable area? Or sorry, water is pretty polar. So why did I put it down here by the permeable area? Water is very small. And we're going to see later in our lesson that water actually, even though it's polar because of its small size, will cross through the membrane. So we have some molecules, things that are large in charge, very impermeable. We would also call these, a lot of times we were in the medical community, we also would call these non-penetrating molecules. And then over here, we would have penetrating molecules. Okay, and so we just have some things that cross the membrane all on its own and other things that are impermeable and don't cross the membrane. Now, let's come back over here on this side, and we're going to draw another membrane. And we are going to talk, now that we've learned that some things are permeable or penetrating, let's label this as our membrane again. Now we're going to talk about a process called diffusion. And what we call concentration gradients. And so a concentration gradient exists inside the cell anytime that we have a membrane and we have a solute. So right here, let's draw some red dots. And this is going to represent our solute. And anytime we have a different concentration of solutes across a membrane, we have what's referred to as a concentration gradient. Concentration gradients are significant inside the cell because they can be a source of energy. in our last unit, when we talked about free energy, we learned that inside the cell, there are really two forms of potential energy. One form of potential energy was in the bonds of the molecules inside the cell. But the other form of potential energy was what we called concentration gradients, and then we just left it there. Well, we're going to pick that up again right here. Because the rule of concentration gradients is that gradients will flow from a high concentration. So up here, we have a high concentration of the solute. Remember, we use these square brackets to signify concentration. So we have a high concentration on that side, and we have a low concentration of solute on that side. And what will happen, due to entropy, we're going to get a flow or a movement of that solute from high concentration to low concentration. And that just happens naturally. It's like water flowing downhill. Okay? So things will move from a high concentration to a low concentration. The membrane as a barrier will stop them if it can, but if they're permeable to the membrane, then they'll just flow. We can actually, in some cases, if they're not permeable, like an ion, then we can create some kind of channel or pore that they could go through and get across the membrane. And then this becomes, we can actually, as they flow from high to low concentration, we can actually release energy. So the movement of solutes, what we call down, so we call this down, or with the concentration gradient, releases energy. Now likewise, we could actually move things, so we don't have to move it from high to low, but that does release energy. We could also take a gradient and move that gradient from a low concentration up to a high concentration. So now we're moving from low concentration to high concentration. And as you might guess, that is called moving up. Or we would also say it's moving against the concentration gradient. Well, if we released energy moving down our concentration gradient, then when we move up our concentration gradient, then this requires energy. And so that's what a concentration gradient is. Now, so far, we have only been looking at a solute that has essentially been neutral in charge. There's been no charge to it. So let's, on this far side here, extend our understanding of concentration gradients to a new term called an electrical gradient. Now, to understand electrical gradients, we have to understand a few things about the cell. And the first thing that we have to understand is that the membrane, across the membrane, there exists a separation of charge. And that is referred to as the membrane potential. So we'll just say that is a separation of charge. And what that means is the inside of the cell, we'll call this bottom layer right here, we'll call it the inside of the cell, or the cytosol. And so out here is the extracellular matrix. And what happens is the inside of the cell, it turns out inside the cell that there are more negative charges inside the cell than there are outside of the cell. These negative charges are coming from negatively charged phospholipids. that line the surface of the plasma membrane, but only on the cytosolic leaflet. They also come from the fact that you have a lot of phosphate ion inside the cytosol and not outside. A lot of the proteins that exist inside the cytosol overall have a negative charge on them. And so all of those combined together produce more negative charges inside the cell than outside the cell. And that's a separation of charge. The inside of the cell is more negative than the outside of the cell, and that's what the membrane potential is. Now, if you're a neutral molecule, like this red circle that we have up here, you don't care. If you're neutral, you don't feel the charge anyway, so it makes no difference to you. But if you are a charged solute, like, let's say, a sodium ion, then you have two gradients that you're working with. So now we've got sodium ions up here. And what's going to happen? Well, as we drew over here, there is a concentration gradient, and so sodium ions are going to want to come in. We'll use that C to represent concentration. But there's another force at work here. Because the sodium ions are positively charged and the inside of the cell is negatively charged, then the positive charge is going to have an additional force pulling it inside of the cell to get to that negative charge on the inside. And so we call that the electrical gradient. So a positive charge on the outside of the cell has an extra draw force pulling it inside the cell. Now, in this case, these two forces, we call them together the electrical or electrochemical gradient. And let's write that word down. Electrochemical gradient. And the electrochemical gradient, again, is the sum of these two forces, the concentration gradient, which in this case would pull the solute inside the cell, and the electrical gradient, which is pulling the solute, because it's positively charged, into the cell. So in this case, the two forces are combining to work with each other. So they're summative, they're adding to each other. That's not always the case, though. We can have another ion. So let's draw another common ion inside the cell is potassium. And potassium is also positively charged, so we'll make this green circle equal to potassium. And so now, with potassium, we see that the concentration gradient is actually going to push. These ions are more concentrated. They're at a high concentration. So they're going to want to move, if we provide a protein carrier, they're going to want to move to the outside of the cell. They're going to move in this direction. That's what their concentration gradient is going to tell them to do. However, they're positively charged. So the electrical gradient is still moving in this direction. Here's the electrical gradient right there. So the electrical gradient is going to actually be pushing them into the cell. And so now these two forces are actually working against each other. And eventually they'll cancel out. This might flow to the outside until it's balanced by the electrical gradient pulling those ions back into the inside. And so here they're summative, here they're subtractive. They're working against each other. You could also figure out, we won't do it, but imagine what might happen if you had a solute that was negatively charged, like chloride. And that might be something to think about. What impact, if you were to draw a gradient with chloride ions, what would happen with it? But we'll just leave off right now saying that gradients exist inside the cell, and concentration gradients can work as an energy force. And they will move from a high concentration to a low concentration. We call that moving down your concentration gradient. If you add some energy to the system, you can move from a low to high. That concentrates things. We do this inside of the cell. We'll pull nutrients from the outside and bring them inside the cell to concentrate them. But it requires energy. And then we also learned that in addition to just the concentration gradient, we can have a second force that acts on charged solutes. And that's the electrical gradient. Together, they're called the electrochemical gradient. Now, we've been learning a little bit about gradients and diffusion. And we're going to come back over here to this side of the board. And we're going to talk now about another process called osmosis. And osmosis is going to focus on this particular solute, water. So now we're going to talk about osmosis. And osmosis is related to diffusion and concentration in that osmosis is defined as the movement of water down or with its concentration gradient. Now this can be a little bit counterintuitive because generally when we think about a solution and you've got all these solutes, you might have glucose in there, you might have amino acids. So we've got this solution and we don't think of water as really being a solute or having a concentration. It's everywhere in there, right? And so it's a little bit counterintuitive to think that water can move down its concentration gradient. So to understand that process a little bit better, it helps to visualize what we call an osmotic chamber. And so we're going to just draw up here on the board a chamber or a tank, and it's going to have two chambers, A and B. And we're going to fill those with water. And we're going to assume, I know I probably haven't drawn this up perfectly, but we're going to assume that both of these chambers are exactly the same volume. And then we are going to put a membrane in the middle here. So this is our membrane. And it's going to be permeable to water, but impermeable to whatever solute we put in these chambers. And so now let's add a solute, and I'm going to take and add some solute. And now ask yourself the question, if you look at this model, remember that the volume, the level of water, is the same in both of these chambers. Volume's exactly the same. But as you look at these, ask yourself the question, does one chamber, A or B, have more water in it than the other chamber? And I'll give you a second to consider. Or do they both have the same amount of water, or does one have more water than the other? Now, as you start to look at that, hopefully you arrive at the same conclusion that I do, and you would say that chamber A actually has more water. So this one has a higher concentration of water. Why? Because the total volume here is made up of two things, the volume of water and the volume of all these green solutes or particles that we put in there, right? So, if the volumes are the same on each side, this one has fewer solutes and therefore must have more water. Over here we had more solutes, and so consequently on this side we would have less water. So now let's apply what we learned in our last segment on diffusion. Remember, diffusion molecules always flow from high concentration to low concentration. Well, here's the high concentration of water. Here's the low. So water, by osmosis, water is going to move across a membrane down its concentration gradient. And that's what osmosis is. So I'll write osmosis on top of that arrow right there. So osmosis is going to cause water to flow from its high concentration to its low concentration. Now, that water will continue to flow until what it's trying to do is equalize the concentration of water. We could also, it's a little bit hard to visualize the concentration of water. So it might be easier to think that the water is going to flow here until the concentration of green solutes is equal on both sides. So it will just go until it hits that equilibrium, or that's what it's going to try to do. As osmosis pulls water from chamber A to chamber B, we are going to get the level of water in chamber A is going to go down as water moves out of it, and the water in chamber B is going to move up. And so we can actually change the water levels. And in fact, that water will keep flowing until the concentration is equal. Or the other thing that can happen is, remember on this chamber, we've got things like gravity that are going to be pushing down. There are some chambers that we actually put a piston on top of it with a gauge. We might have gravity, or we could, let's just draw some kind of piston. And then we put a gauge on this. And we can measure what this will do is that it pushes up against the piston. It will create a certain pressure in here. So the movement of water can actually create pressure or force. And we would call that force the osmotic pressure of it pushing up against there. And so that's what osmosis is. We could write this down as a general rule that water always flows towards the region or area or space chamber the chamber of high solute concentration. Okay? And so that's a rule. It's just looking at the opposite. We're still saying it's flowing down its concentration or toward less water. But anytime you have a higher solute concentration, you're going to have less water. And so the general rule is that water always flows towards the region, the chamber, the space that has a high solute concentration. Now, let me change one other feature. So let's come over here. And I'm going to erase this model. I'm going to draw up a new model. I'm going to change my chambers. I'm going to leave all the green dots exactly the same. Let's put our levels of water back up so that they're equal also. And we'll redraw our membrane. And now, all I'm going to change is I'm going to add another solute. We're going to add some purple squares as another solute. All right. Now, and we're going to use this model to illustrate an important concept with osmosis. Over here, Before, we said that osmosis was going to move water in this direction because we have more green dots over here than we did over here. Now, I changed it a little bit. The green dots haven't changed, but I've added purple squares to one side. Will osmosis drive water in the same direction or a different direction? And as you start to think about that, notice that I didn't say any particular solute concentration. Water will flow toward wherever the largest total solute concentration is. So in this case now, we're going to have, because there are more total solutes, it doesn't matter whether they're purple squares or green circles. There's more solutes over here total, and so now osmosis is going to drive in this direction because this has the higher total solute concentration. And so that brings up another important rule about osmosis that we should put up here, and that is osmosis follows total solute concentration, and it ignores solute identity. So it doesn't care whether you're glucose or sodium or chloride. A particle in that solution is a particle. An ion is an ion. A solute is a solute. And so you have to add up all the solutes. And so this brings us to a new term that we want you to know, and that is osmolarity. Osmolarity sort of sounds like another word you know, molarity, and that's where it comes from. Molarity, remember, is an expression of the concentration of a particular solute, a concentration of a particular solute. So for example, you could have 1 molar glucose. You could have 1 molar sodium chloride. You could have 1 molar potassium chloride. Those are all concentrations of a single thing, a single type of solute. Osmolarity, again, we're going to take molarity or concentration, but combine it with osmosis. And we're going to say osmolarity is the concentration of total solutes, or I often say of total particles inside of that solution. And the reason why I say that is because let's say we earlier said an example. We said you could have 1 molar sodium chloride, right? So we take 1 mole of sodium chloride, we dump it in our water, it would dissolve. We've learned earlier that sodium and chloride are held by an ionic bond that when it hits water dissociates. And so it actually produces 1 sodium ion and 1 chloride ion. So if we start with a 1 molar sodium chloride, what would be the osmolarity of that solution? And this starts to point out we now have that 1 mole of sodium chloride broke into 2 different particles. So even though it was 1 molar sodium chloride, that solution is going to be 2 osmolars, right? Because the sodium ions are contributing 1 mole of particles, and then the chloride are contributing another mole of particles. So if things dissociate, then it gets an increase in the osmolarity. Other things, like if you take glucose, glucose is made up of carbon, hydrogen, oxygen. It is made up of all covalent bonds. They don't dissociate in water at all. So when I add 1 molar of glucose solution, I get 1 osmolarity, or 1 osmolar solution. Because the glucose, every 1 glucose that went in stays 1 glucose. It's 1 particle. Inside the cell, we won't talk about everything that's inside the cell, but it can get kind of complicated. For example, inside the cell, we might have 1 molar, well, we wouldn't have 1 molar glucose inside the cell, but let's say we had a solution that was both 1 molar glucose and 1 molar sodium chloride. And just to be fun, we'll say 1 molar potassium We'll just call it potassium chloride also. What would be the osmolarity of that solution? Well, to figure out the osmolarity, we would have to add up the contribution from each of those. Remembering that these two are salts that will dissociate. So they're each going to, this will form 2 particles for every 1 particle of salt, and this 2 particles for every potassium chloride. So our total would be 5 osmolar for that particular solution. Okay? And so that's osmosis, and we've talked about this idea of osmolarity. We've been now able to predict which way osmosis will flow, and understand better an osmotic chamber and things like osmotic pressure. So now we're ready to talk about osmosis and diffusion and its effects on the cell. So now we're going to take what we've learned about osmolarity and we're going to start applying that to cells. And how does osmolarity impact cells? And so we had to learn about a couple of, one more new term. So we're going to continue talking about osmolarity a little bit. And then we also need to introduce a new term called tonicity. And so in this case, what we're going to be doing is we're going to be considering, I'm just going to draw a chamber here, and then in the middle, I'm going to draw this round circle. That's going to be a cell. And then, so this cell is being bathed by whatever fluid is in this chamber. So we really have 2 fluids. We have what's called the intracellular fluid, or ICF. And then over here, we have the extracellular fluid, or ECF. And we just learned that you have osmolarity, so you can have inside the cell and outside the cell, can have different concentrations of solutes. And if they are different, then you can get osmosis or movement of water across that membrane. And that can have an impact on the cell. So we need to give you a couple of terms to be able to discuss these types of situations. And the first set of terms have to do with osmolarity. So we have a term called hyperosmotic. Okay? So hyperosmotic describes the osmolarity of a solution in relationship to another solution. It doesn't have to be the ICF and ECF. Here's 2 solutions. It could be 2 beakers next to each other. And the hyperosmotic simply means that one of the solutions is hyperosmotic or has a higher concentration of solute, whatever that might be. And that comes from the fact that hyper, where I remember this, is hyper means elevated or raised or increased. Right? We use it in other words. It's a common Greek prefix. So we use it in words like hyperactive. So we all know that hyperactive, if you have a hyperactive child, then that child is bouncing off the walls. They're really active. They're elevated activity. The opposite of a hyperosmotic solution would be a hypoosmotic solution, where hypo is also another, it's the counter Greek prefix, and hypo means decreased or lowered. We also see hypo in some common words in our society. For example, you may know someone who had hypothermia before, or a decreased or lowered body temperature, hypothermia. You may have gone to the doctor's office and been stuck with a hypodermic needle. So that means that the needle, hypo, goes beneath the dermis, or skin. Underneath the skin is what hypodermic literally means. And that's where that term comes from. So if you can remember some of those common words, the reason why I tell you the Greek prefixes and what they mean is because these both start with high and end in tic, and it's really easy to confuse hyperosmotic and hypoosmotic. And so you really have to think, okay, what's that prefix mean? But hyperosmotic is then going to be the solution with the lower solute concentration. And then there's one other term, and that's going to be isoosmotic. And when you have a solution that's isoosmotic with another solution, that means that iso means the same or similar, and so that means that they have the same solute concentration. And so now we have set up, if we have the cell sitting in some kind of fluid, we have terms that we can describe the osmolarity of this external fluid, or we could say the internal cell fluid also. So if we drew some dots, so I'm going to draw a representation of 3 red dots, and those red dots are going to represent all of the different solutes that might be in that cellular fluid. Obviously, there's a lot more than 6. I'm just using that as a representation. And so we have 6 dots inside the cell. And outside the cell, I'm just going to draw some purple dots to represent extracellular solutes. And now as you look at this, and let's assume that the 2 compartments have the same volume again, is one of these fluids, the ICF or the ECF, have a greater osmolarity than the other one? Does it have a greater total number of solutes? Well, outside has 8 solute representations, inside has 6. So hopefully your answer is yeah. One of these has more solute, and it's the extracellular fluid. That would mean that the extracellular fluid would be the hyperosmotic fluid. The intracellular fluid would be the hypoosmotic fluid. If I remove some of these dots, so now I've got 5 and 6, then that switches. Now the intracellular fluid is hyperosmotic. It has the higher amount of solute. The ECF is the hypoosmotic. It has fewer. And if you drew the same number of dots, then they both would be isoosmotic. Now, the reason why that's important is because the amount of solutes that you have inside and outside the cell regulates osmosis, or the flow of water. So water will actually flow into the cell or out of the cell by osmosis based upon the osmolarity in that cell. And we have some other words to describe that, and that's where tonicity comes into play. So let's talk about tonicity for a second. Tonicity comes from the place where we're kind of more familiar with this word of tonicity. When you think of tonicity, I want you to think a little bit, we often talk about someone having good muscle tone, right? And so when someone has good muscle tone, that means that their muscles are firm, right? And so tone has to do with the firmness of something. Now let's stop thinking about muscles in terms of tone and start thinking about cells in terms of tone or firmness. Or if you want to imagine like a balloon in terms of firmness, and you can really swell that thing up, or maybe a rubber ball in terms of firmness. You can really have it swollen with air, and so it's really firm, or it could be kind of deflated and get a little bit squishy, right? And so when we talk about tonicity, that's what we're talking about, the firmness of the cell. And just like a ball, instead of filling it up, with a ball we might fill it up with air, but instead of filling it up with air, we're going to be filling it up with water. And so the tone of that cell is a function of how much water is in that cell. And so tonicity, as we refer to these tones, it refers to the firmness of the cell. And then we know that solutions can affect that tone, because different solutions can affect which way osmosis goes. And so we start to get some different terms. So for example, you can have what we call a hypertonic solution. Okay? Now we already talked about hyper. Tonic is a new suffix or root word up here. If you put the cell in a hypertonic solution, then water flows out. Okay? So the water is going to flow out of the cell, and the effect on the tone, or the effect on the cell, is going to be that that cell is going to shrivel or shrink. And so this is the effect. This is the direction, and then this is the type of solution. A couple of other things that we want to know about a hypertonic solution. In animal cells, this creates a condition known as acrenylated or crenation, which means it gets shriveled up. It looks kind of like a raisin. In fact, that's pretty much the same thing that happens to raisins. You have a nice, firm, plump grape with good tone, and then you dehydrate it, all the water comes out, and you get this shriveled or crenated raisin. So if you want to, you can put in parentheses, this is your raisin. That's what it looks like. Now, in plants don't get raisin shaped. The plant cell actually has that cell wall around it, so it actually stays the same size, but you do get something called plasmolysis. And in plasmolysis, so here's the plant cell with its cell wall right here, but the plasma membrane inside starts to tear away from the cell wall a little bit. So it's almost like it forms a raisin inside of a hard shell. Okay? And so this would be what we call plasmolysis. So that's a few words, associations that we want to put together with this idea of a hypertonic solution. If you put the cell in a hypertonic solution, water flows out of the cell, it shrivels. In animal cells, this is referred to as crenation, and in plant cells, we call it plasmolysis. In almost all cases, it's bad for plants and it's bad for animals. You could also have then, we learned that the opposite of hyper is hypo, so you could have a hypotonic solution as well. As you might guess, in a hypotonic solution, water flows in to the cell. Now, instead of shriveling, that cell is going to swell or become bigger. In animal cells, they don't have a cell wall, so as water moves in, just like forcing too much air into that ball or that balloon, it leads to a condition called lysis, which means the cell eventually pops. So hypotonic solutions are not good for animal cells. It causes them to pop and lyse. In plant cells, however, they do have a cell wall. And so the plasma membrane swells up against that cell wall, and you get a condition known as turgor. We'd say that those cells are turgid. And that's actually a good condition. So plants prefer to be in turgor. Okay? They like that. And so a hypotonic solution is actually ideal for plant cells. Not ideal for animal cells, they pop. So hypertonic solutions are good for no one. Hypotonic solutions are good for plants. Hopefully there's one good for animals in here, right? And there is. The last one is you can bathe that cell in what's called an isotonic solution. In an isotonic solution, there is no water flow, or at least no net water flow. There's some water molecules coming in, but there's an equal number coming out. So there's no net water flow. So what's the effect on the cell? Well, there's none. It stays at whatever tonicity it was when it entered into that solution. In animal cells, we just call this normal. And in plant cells, it doesn't actually start to, we call it flaccid. And so the plant cell stays the same, but this is, the cell is not swollen, it's not pushing up against the plasma membrane, so it looks a little bit like those bouncy houses when you start to let a little bit of air out and they just become more flaccid. You may have noticed this at the end of the semester, for example, when you go to clean out your refrigerator and there's that bundle of celery that your roommate left there from 2 months ago, and you kind of pull that celery out and you can kind of go like this and the celery kind of wobbles like that. That's because those celery cells have lost a lot of their water and they're in this flaccid state. Now, if you go ahead and take that celery and you put it in a hypotonic solution, let's say you put it in some distilled water, then that water is going to move across into those cells, and then you'll create a turgid state, and then that celery will become firm and crisp again. Okay? All due to the movement of water. So, we've now learned about osmolarity and how we can compare solutions based on the cell. And we've learned about the effect, or the movement of water, and what effect that has on the cell, particularly its tonicity. Now, let's learn a couple more things and then apply this to some clinical settings of what might happen and how you might manipulate the tonicity of cells using solutions that you could bathe the cells in. Okay, so now let's look at the application of these concepts of osmolarity and tonicity in a clinical setting. And so there's a couple of things that we need to know, just some basic information, in order to understand these applications. The first, we talked about osmolarity before. It turns out that most cells have a common osmolarity, and so the osmolarity of most cells, so we would say the intracellular fluid, is equal to 300 milliosmolar. Okay? Now, two other pieces of information. We are going to, often in the clinical setting, so in a hospital, they're going to, if you've ever been in a hospital maybe for surgery or something and you went in, and they probably gave you an IV or an intravenous line that was hooked up to a little bag on a hanger. And that little bag probably contained one of two different solutes. One of them, or one common solute, is called dextrose. Dextrose is the medical term that we use for glucose. So you already know what dextrose is. We've been talking about it all class long. And it turns out that dextrose, if you prepare what we call a 5% solution of dextrose, then that also equals 300 milliosmolar. Now, one thing important about dextrose, if you remember way back to the beginning of this lecture today, dextrose, or glucose, is permeable to the membrane. We would call that penetrating. You may argue that it doesn't penetrate really well, but it does penetrate. So let's remind ourselves of that. And we're going to say that this is a penetrating solute. The other common solute or solution that you're going to find is called a saline solution. Now, a saline solution, what they call a saline solution in a medical setting, chemists and biologists just know as a solution of sodium chloride. And as we saw with glucose, we can also prepare a concentration of saline that is equal to 300 milliosmolar. That's going to be a 0.9% saline solution. So, with that information, we are ready to tackle our first problem. And so what we're going to do, we're going to use just a chamber to understand this concept, much as like we drew previously. And so we're going to have a cell. This could represent a whole organism or all the cells, but we're just going to make it simple with a single cell. And that cell is being bathed in a solution. Now, what kind of solution are we going to have? Let's start out, let's consider a 5% dextrose. So we're going to surround this with a 5% dextrose solution. Now, let's first start by representing in this model, as we did earlier, I'm going to draw 6 dots. And those 6 dots are going to represent my 300 milliosmolar osmolarity inside the cell. They're representing all the proteins, the phosphates, the ions, the nucleic acids, nucleotides. Everything in together is represented by those 6 red dots. Okay? That big conglomeration of all those different solutes. Now, on the outside, I have a 5% dextrose. Well, if I represented 300 milliosmolar inside the cell with 6 dots, then I should also represent the 300 osmolar outside of the cell with 6 dots also. So we'll draw 6, we'll represent those with a purple dot. And so we're going to draw 6 purple dots here representing that solute. Now, the first question is, what is the osmolarity of that solution? So osmolarity. And then we want to know and understand what is the tonicity, or what's the effect on the cell of that solution. Now, to do that, so we put that cell, we bathe that cell in that solution, and then something's going to happen. Some things are going to move. We have to first consider the effect of diffusion. And then after that, we have to consider the effect of osmosis. So, let's redraw this chamber and look at those 2 effects. So this was a cell at our initial conditions, right when we dropped it in. And then after diffusion and osmosis occur, what does it look like? Well, here's our cell. We had the red dots inside. Do they diffuse at all? And the answer to that is going to be no. If they were diffusing, they would have diffused a long time ago. So everything that's inside the cell stays inside there. All the proteins, the DNA, the ions are too big or too charged to move outside of the cell. But we've got glucose on the outside. So we ask ourselves, was glucose able to diffuse? Well, remember earlier we said that it's penetrating. And so, yeah, glucose diffuses. We had 6 dots. But these dots will diffuse across that membrane, and so they do so. Now, how much do they diffuse? Well, they're going down a concentration gradient. There's no glucose initially. There's 6 on the outside. So they'll diffuse until the concentration of glucose, not dots, just purple dots, is equal in both chambers. That's what diffusion does, right? It moves down its concentration gradient until there's no longer a gradient, until they're equal on both sides. So we're going to have purple dots move inside the cell until the amount of purple dots is the same on each side. Why is that happening? Because it can, because it's penetrating. So we just considered the effect of diffusion of those solutes. Now let's look at osmosis. What does osmosis do now? Well, we learned that osmosis moves water to the higher concentration of solutes. Do we have a difference in concentration of solutes? Yeah, we do. Water in this case, I've got 9 dots inside. I've got 3 dots outside. So water is going to move inside that cell. When that water moves inside its cell, what's going to happen to it? Well, we learned earlier that as water moves inside, it causes that cell to swell. That means that a 5% glucose solution, dextrose, that we set up earlier, what's its tonicity? Well, it was going to cause the cell to swell and probably eventually lyse if it's an animal cell. So 5% dextrose must be hypotonic. On the other hand, when it first started out, it was the same concentration outside the cell, 300 osmolar, as inside the cell, 300 osmolar. So the osmolarity was actually isosmotic. Now, one more thing before we go on to our next problem. One of the things that can be hard to remember is the hypotonic and the hypertonic is not quite as clear as the hypo and hyperosmotic, right, of which one's greater or lesser. So one of the tricks that I use to remember which one goes with which effect is hypo ends with the letter O. And if you think of an O as being a big swollen cell, that O, then that is really how I remember that hypo is associated with cellular swelling, that big O shape. And then I just remember that hyper must be the opposite or the shrink state. So that's how I remember that hypotonic is associated with the swollen or lyse state. Now, that's how you would do and set out these problems. So we know if we give someone a 5% dextrose solution, that is going to be hypotonic or cause water to move inside of those cells and pull it inside. Okay? Now, let's consider a different solution. So let's change these. We'll leave the cell the same. Now, let's look at a 0.9% saline solution. So we'll represent saline with green dots. Now, from saline, sodium chloride, over here we said earlier that dextrose was penetrating. Saline is non-penetrating, which means it won't go across that membrane because it's charged, sodium and chloride ions. So let's represent this. Again, we've got 0.9% saline. That's 300 milliosmolar. So I'm going to represent that in my model with 6 green dots. And let's erase these. Let's ask ourselves the same question. Osmolarity under my initial conditions, what is the osmolarity? Well, I got 6 dots on the outside, 6 dots on the inside. So this is actually isosmotic. Now, what about the tonicity? What effect will this solution have on the cell? Well, let's go back to our arrow. First, is there any diffusion? Well, we have to draw, I guess, our green dots over here also. And the first question we ask, is there any effect from diffusion? Well, these ions cannot diffuse across the membrane, so there's actually no effect. So we've considered diffusion, and we said that doesn't happen because saline is non-penetrating. So the red dots can't penetrate outside, the green dots can't penetrate inside. So after diffusion, our model looks exactly the same. Now, how about osmosis? Will water flow in one direction or the other? Remember, water will always flow toward the higher concentration of solutes. We've got 6 solutes on the outside in the ECF. We've got 6 solutes on the inside in the ICF. And so there's actually the same number of solutes after diffusion. And so osmosis has no effect in this case. There's no net movement of water, which means that there's going to be no effect on the cell. That was our definition of an isotonic solution. An isotonic solution is one that has no effect on the cell. So 0.9% saline has no effect on the cell. So if you're in a hospital setting and you want to just hydrate someone, give them some fluid, but not pull water in or out of that cell, then you would consider giving that person a 0.9% saline solution. Because that's going to keep, if you put that in the blood, it's going to keep the cells happy. Water isn't going to move out of the cells. Water's not going to move into the cells. Now, we've looked at these 2 different solutions, dextrose and saline, individually. A common thing that comes up is, right here we saw that the osmolarity was iso. It matched the tonicity. We had isoosmotic, isotonic. As you do these types of problems, a lot of times students want to say, oh, is it always the same? Is isoosmotic always isotonic, hypotonic, or hypoosmotic always hypotonic, and so on? And the answer is no. It depends on what your solution is. Remember, in our first example, we saw that they were different. Isoosmotic dextrose actually became a hypotonic solution. In the study guide that you guys have, or the worksheet that you guys have, and in the hospital clinical setting, so far we just took the isoosmotic concentrations of dextrose and saline. However, you can get different concentrations of dextrose and saline and do different things with the osmosis to the tonicity of cells. So what would happen? You might do some problems where you ask yourself, what would happen if instead of 5% dextrose, I had 2.5% or 1/2 the osmolarity? What if I had 10% or double the osmolarity? What if I had 0.45 saline, half the osmolarity? What if I bathed it in 1.8 or double the amount of saline? What if I started to combine? What if I put saline and dextrose into the same IV bag, the same solution? In each case, what effect, what will that have on the cell? Will that be a hypotonic, hypertonic, isotonic solution? And so these are good activities to think of. And as you do some of those different sort of thought experiments, see if you can identify some patterns that occur. And so we'll leave off our lecture with that, being able to do, so this is a skill set, being able to solve this type of clinical application problem. And you'll find some prompts in the worksheet as you go explore that. Thank you.